Hello and welcome, dear friends. In my last lecture, we saw the basic aspects of the nitrogen cycle. Today we will be discussing the microbiological aspects of this nitrogen cycle and also a bit of enzymology which is involved along with the microorganisms associated with it. We have seen that this is a very important part of the biogeochemical cycle, and the one of the element which is continuously recycled, and it forms a vital part of the energy flow and the nutrient recycling which takes place along the food chain. So the producers, they are at the very key level of the food chain, and they are the ones who are constantly involved in uptake of this atmospheric nitrogen, and with the help of microorganisms of course, and then transfer it to the next level of the consumers, that is the primary, secondary and the tertiary consumers. Once the tertiary consumers they die, they are ultimately subjected to soil, where the microbial transformations, that is what we call it as the decomposition or the decay, takes place. And by this, just like the money in the market, nitrogen is continuously recycled, transformed from one form into the another, and the net balance is maintained. So first a few slides I would like to explain it through a question and answer form, that is through some questions which may be there in your mind, like where is nitrogen found in the environment, why this nitrogen is needed to be transformed, how this nitrogen gets transformed. There will be many questions associated with nitrogen cycle in your mind which needs to be answered. So I have chosen a way of inquisitive method of teaching. In inquisitive method, you have a question and then there is an answer. So as this is a online video lecture, I would be dealing with the question and I myself will be answering it. So I hope you enjoy this particular mode of teaching. Later on in the second part of this lecture, we will see the 5 steps which form this nitrogen cycle. So let's begin with the question, where is nitrogen found in the environment? The largest single source of nitrogen is the atmosphere. It makes up 78% of our air. Apart from this, nitrogen is present in soil in the form of nitrites, nitrates, ammonium salts, etc. These compounds are also used as fertilizers or to fertilize the soil so that the plants they can uptake it. Nitrogen, when it is uptaken by the plant, it gets transformed into a much more complex macromolecule like proteins, and these proteins, these are a part of become a part of the living system. So nitrogen is found in proteins and other organic compounds that are present in meat, egg white, vegetables, etc. So it goes on to become a part of the biomass. The second question which may be there is, what happens to the atmospheric nitrogen in the nitrogen cycle? As we have just seen, 78% of the air, it contains nitrogen. What happens with this? Atmospheric nitrogen is converted into ammonia or nitrates. Atmospheric nitrogen is converted into ammonia or nitrates. This is the fate of atmospheric nitrogen. We have seen lightning may be there. There are certain free living bacteria, some bacteria which get associated with it. What are the names of this bacteria? That will form the part of the second part of this same lecture. So first try to understand or answer your basic queries. The atmospheric nitrogen is converted into ammonia. Nitrogen combines with hydrogen or oxygen. Okay, so when it combines with hydrogen, it forms ammonia. When it combines with oxygen, it forms nitrites and nitrates. There are a specific group of bacteria associated with it. We will see them one by one. Now, why does atmospheric nitrogen need to be converted? Why there is a need for the atmospheric nitrogen to get converted into ammonia, nitrites, nitrates, whatever it is? Now this question is just like, why do we need to convert the money into gold, or gold money into land, or money into food. There is a circulation, economic circulation in the market. If you see the commercial aspect of human life, mostly our currency is money, and we need to transform it into one form into another for living. Just like this, the nitrogen, atmospheric nitrogen, need to be converted. It is one of the nature's greatest ironies that nitrogen is such an essential compound, but we cannot use it. It is an essential component of DNA and proteins, the building block of life, and the majority of the air which we breathe in is nitrogen, but we are unable to use it. The most living organisms, not only we but most of the living organisms, they are unable to use the nitrogen as it exists in nature. To compare this, we can use oxygen as it is from nature. We don't need anybody to fix it. We have developed our organs in the form of lungs, in the form of hemoglobin in the blood, the iron which carries the iron moiety in the hemoglobin, which is capable of carrying the oxygen. So these evolutionary modifications are not there with respect to nitrogen, and nitrogen can only be fixed by bacteria. This is the beauty part of the whole nitrogen cycle, is the key players are microorganisms, and indirectly they are helping us, plants, animals, to obtain the atmospheric nitrogen inside our body. So we are assimilators. We are the part of the assimilation process in nitrogen cycle. I'll come to that, but there are, let us say, producers or nitrogen fixers, there are assimilators, then there is a dissimilation of the nitrogen, and it is given back to the environment. So that is the whole point of the story. Now next question, how does atmospheric nitrogen get changed into a form that can be used by most living organisms? How does this atmospheric nitrogen gets converted? Now there are four main ways by which there is transformation of this atmospheric nitrogen. Each of these ways has its own oxidation state, because all these are redox reactions, the oxidation reduction reactions. Now by traveling through one of these four processes in the nitrogen cycle, the atmospheric nitrogen is changed into a form which can be used by us or other living organisms. The first is nitrogen fixation. The second is ammonification, formation of ammonia from the atmospheric nitrogen. Third is denitrification, that is conversion into nitrites and nitrates. And the fourth is denitrification, where ultimately it is lost or given back. Rather than saying lost will, I think it will be a selfish terminology to say is lost in the environment. Actually it is given back to the environment. So these are the four stages by which the nitrogen moves in the atmosphere. So what is nitrogen fixation, and what does it mean to say nitrogen gets fixed? This should be the basic question in the empirical mind of a science student. What basically is nitrogen fixation, and what does it mean to say when you say that nitrogen is fixed? The first process in the nitrogen cycle is nitrogen fixation. Nitrogen fixation is the process that causes the strong two atom nitrogen molecules, which is bonded by 3 bonds, to which is found in the atmosphere, to break apart so that they can combine with other atoms like hydrogen and oxygen. So this is nitrogen when it is broken apart. How is it broken apart? That should be another question. How does this nitrogen, 3 bonds of nitrogen, is broken apart? It is broken apart by lightning, but that is mathematically speaking a very less quantity but very significant one. The second is by microorganisms, that is by enzymes. I will come to that in the explanation when I will explain it step by step. So nitrogen fixation is a process that causes the strong 2 atom nitrogen bonded by 3 bonds found in the atmosphere to break apart so that they can combine with either oxygen or hydrogen to give rise to amines or nitrates. So this is nitrogen fixation, the first part. Nitrogen gets fixed when it is combined with oxygen or hydrogen. This is the basic first step in nitrogen cycle. Which are the organisms? We will see it afterwards. Now, having understood this, there will be another question. There are how many ways are there by which this nitrogen can get fixed? Is lightning the only way? Is microorganism the only way? There are certain ways, by 2 to 3 ways by which nitrogen can get fixed from the atmosphere. Now, nitrogen is present in the atmosphere, it is present in the soil, it is present in the oceans, so on and so forth. So all this need to get fixed in their own perspective. So if you have to see a bio-geo physico-chemical perspective of nitrogen cycle, then to be very broad in perception, I'll have to say that the land nitrogen cycle is different, the ocean nitrogen cycle is different, the soil nitrogen cycle is different. Even though if it's different, it is different in terms of geological perspectives. It is same in terms of the biochemical perspective. Means the biochemistry of nitrogen cycle remains the same. But geospatial terms, the players will be different, the ground is different where they are trying to fix it. So that is a part of the broader study which you can explore into. But there are 3 ways by which nitrogen can get fixed. First is the atmospheric fixation, industrial fixation, and biological fixation. Atmospheric fixation, only 5 to 8% of the fixation takes place by lightning. Nitrogen combines with oxygen, nitrogen oxides are formed, nitrogen oxide dissolves with the rainwater, and nitric acid HNO3 is added as acid rains into the soil. So this enormous energy of lightning breaks the nitrogen molecules apart, enables the nitrogen to combine with oxygen, forming N2O or nitrogen oxides. Nitrogen oxides dissolves in the rain and forming nitrates. Nitrates are carried to the ground, and this is one of the way by which nitrogen is fixed. As I say, this is a very important natural process which is going out in the nature. Now the second is industrial fixation. Industrial fixation, first we saw atmospheric fixation. Now the industrial fixation, it is done by some processes called as the Haber's process, where nitrogen is formed, or there can be formation of fertilizers, ammonia fertilizer, under great pressure at around temperatures of 600 degrees centigrade, and with the use of a catalyst like atmospheric nitrogen and hydrogen, they are combined to form ammonia. In the industrial plant, it combines nitrogen and hydrogen to form ammonia. Ammonia is used as a fertilizer in soil. Okay, so this is the second type of fixation which is called as the industrial fixation. The third is biological fixation, which we microbiologists are very much interested in. Biological fixation is the most important complete form of fixation, most important nitrogen fixation, which is the process we say is completed when it is biologically fixed. Okay, so there are 2 types of nitrogen fixing bacteria. Biological is primarily involving microorganisms, so we can say it as a microbiological fixing. There are 2 types of NFB, that is nitrogen fixing bacteria. One which are free living, which are not associated with any plant or the plant organelles, and the other is symbiotic nitrogen fixing bacteria, which are associated with the plant. They form a root nodule. Why do they form the root nodule? There is a very interesting story behind it. And these free living, they form cysts. They have a certain structure on their cell wall, a cell wall like structure. It is not a cell wall, it is a cyst, heterocysts. What are the role of heterocyst? It is serving the same purpose for which the root nodules are formed, to keep an anaerobic environment. So I'll come to that, but this is the third type of nitrogen fixation, that is the biological nitrogen fixation. And there are 2 types of biological nitrogen fixation: symbiotic by the root nodule associating bacteria, and the free living bacteria, non-symbiotic or the asymbiotic type. Okay, so this free living bacteria and the symbiotic, what happens with this? These free living bacteria, these are highly specialized bacteria. As I just said, they have a formation of a heterocyst. They live in the soil and have the ability to combine the atmospheric nitrogen with hydrogen to convert it into ammonia. Ammonia is formed by these free living bacteria which fix the atmospheric nitrogen. So free living bacteria live in the soil, they combine atmospheric nitrogen with hydrogen and change it into ammonia. So 30% of the nitrogen which is fixed is by the free living bacteria. Now the symbiotic bacteria or the symbiotically related bacteria to plants, roots, they have root nodules. There are many plants, they are called as leguminous plants. These leguminous plants, they form root nodule, and these are the structures or the areas where the bacteria they live, they thrive. Bacteria live in the root of legume family plants and provide the plants with ammonia in exchange for the plant's carbon and a niche or a home. They provide the bacteria a habitat, a safe environment to grow. Some of the ammonia may escape in the surrounding soil, enriching it with nitrogen for all the plants. Means a sort of leaching takes place, also ammonia is released and the soil becomes very much fertile. So this is the symbiotic type of relating bacteria which fixes 70% of the nitrogen, yeah, which is acquired by the plants, leguminous plants. Symbiotic relationship bacteria, they fix 70% of the nitrogen. So this is the biological nitrogen fixation. Now most of the atmospheric nitrogen is fixed and it is changed to ammonia. You understood this. But we know that ammonia is highly toxic to many organisms. Ammonia is not that, what you can say, user friendly or consumer friendly. So can plants or other animals use ammonia? The answer is no. Very few plants can use ammonia. So there is fortunately a second step called as ammonification. So very few plants they can use ammonia. I can say it's almost non-significant. It has to be fixed, nitrogen fixation, and second is ammonification. As I said, ammonification now converts ammonia to nitrites and nitrates. So this important step is responsible for converting the ammonia into a utilizable form. So how does that take place? What is ammonification? That should form the next question in your mind. So ammonification is a decomposition of ammonia by bacteria and fungi which break down it into certain compounds like amino acids from dead animals and wastes into ammonia. Bacteria, they break down the decomposers. Now here there are two parts of ammonification. Ammonification is ammonia getting converted into the simpler form in the next step, nitrites and nitrates, and second part is decomposition, decaying. When dead and decaying animals, plants, insects, etc., the biomass, I can say, it is added in the soil, it gets to start decomposed, and this decomposition is carried out by a variety of bacteria and fungi which have the required enzymes for that. Which are the enzymes? We will see it. There is an enzymatic hydrolysis of all the organized molecules in a animal body or a plant body or insect body, and it is constantly decomposed and decays. It is a time consuming process. It requires certain parameters, optimum parameters. All those are present in the soil. So this ultimately, when is complete, it becomes the soil organic matter. So ammonification results into a much more what you can say enriched soil, that is the soil organic matter. So ammonification is decomposition of the dead and decaying into ammonia, or breakdown into amino acids and other animal wastes, we can say. So why is this ammonification necessary and how does this ammonification occur? Now ammonification is not as simple as it is there going out on the surface of the earth. It is a complex process. It can be studied with multiple perspectives. Here I am trying to present you one of the microbiological perspectives. As plants, they cannot utilize the organic forms of nitrogen which are present in the soil, and they are present in the soil as a result of waste or compost, decomposition which I just said, the sludge and the sewage which is added, dead and decaying matter. These all gets converted into organic matter which becomes the soil organic matter, or the SOM. Now microorganisms, they convert the organic nitrogen into ammonia. Ammonia can be either taken up by plants. There are only very few types of plants which can directly take up ammonia, as I just said, in the soil. or it is absorbed into the soil particles. It gets leached into the soil particles. Ammonia in the soil is stored up for a later use, and it is converted into various forms of nitrogen which most plants can use. Bacteria converts the organic nitrogen to ammonia. Ammonia is then stored in the soil, and from the soil, this soil which is rich with ammonia is ready for the next step, that is nitrification, or let us say breakdown. Nitrosification is there. Various different types of bacteria are there which convert it into the utilizable form. Now these plants, when they are ready to take up this broken ammonia, they convert it into another forms of nitrogen like proteins or the nucleic acids, and nitrogen becomes a part of the biomass. So this ammonia which is stored in the soil, what happens with that? As I said, how does it actually get converted into the plant biomass? It travels through a third step now, that is whatever the soil ammonia is there, it travels through the third process in the nitrogen cycle. It is called as nitrification. It is called as nitrification, or the nitrogen conversion of ammonia to nitrites and nitrates. So now let us just run through the general process of this nitrification. What happens with nitrification? Remember, now we are at that stage where atmospheric nitrogen is converted into ammonia. Ammonia is now converted in, is fixed by the, first it is fixed by nitrifying bacteria, then it is converted to ammonia by the dead and decomposing matter, and now the soil ammonia, it has to be converted into a utilizable form which is called as nitrification. So this nitrification process is something like this. This is the overview of nitrification cycle. Nitrifying bacteria in the ground, they first combine with ammonia oxygen to form nitrites. Then another group of nitrifying bacteria. Which are this group of bacteria? What are their names? The further slides it will be explaining all these things. Then the another group of nitrifying bacteria, it converts nitrites to nitrates, which green plants can absorb and use. Nitrifying bacteria in the soil, they combine ammonia with oxygen. We have seen ammonia changes to nitrites first, nitrites NO2, and then again further oxidation results into NO3, that is nitrates. So nitrifying bacteria combine ammonia with oxygen. Ammonia then gets converted to nitrites. Nitrifying bacteria in the soil then convert nitrites to nitrates, NO3. Now plants, they can absorb these nitrates and they can grow. So this is one of the very vital step where the absorption or the transformation of the atmospheric and the soil nitrogen is now given to the biomass nitrogen. So how does nitrogen now re-enter into the atmosphere? This is a very easy perspective which you should understand. Now we have seen the travel of atmospheric nitrogen to soil and soil to plants. Plants, they are the primary producers. Try to understand. There is no such thing called as herbivore, carnivorous, or certain heterotrophs, I can say, which can form or entrap, assimilate their own nitrogen and carbon. No way. Plants are the prime producers of entrapping or assimilating the nitrogen. Only from there we get it. Even if you are drinking milk, even if you are eating meat, or even if you are eating eggs, you should remember that you are getting the nitrogen source from the plant only. How is that so? Even by eating eggs, eggs are coming from hence the poultry, they are eating grains, and grains, they contain nitrogen which has been fixed by the plants, fixed in the plants. So the fourth process, once this is accumulated in the biomass, how long it will remain there depends upon the age of that assimilated system. Assimilation, we humans could have assimilated it by eating the plants. Animals could have eaten it by eating the plants. Other animals, by eating the animals which have eaten the plants, can assimilate the nitrogen. So that is the complexity of the food chain. Now once this all they accumulate and live a certain life on this earth, there is a second stage. now called as the denitrification, where all these animals, they are now ending their life, they die. When they die off, what happens with this is they get decomposed, come back to the stage of ammonification, they are dead and decaying, and this the denitrification process starts. Denitrification converts NO3 in the soil to atmospheric nitrogen by replenishing the atmospheric nitrogen. Okay, what does denitrification do? Denitrification, it converts the nitrates into the soil to atmospheric nitrogen, and it replenishes the atmospheric nitrogen. So this is the nitrogen cycle in general. So this denitrification process, how does this work? Now, as I said, it is the denitrifying bacteria which are living deep in the soil. They may be in the moist sediments, in waterlogged conditions, the swampy sediments, where conditions are very difficult for them to gain oxygen. These denitrifying bacteria, they use nitrates as an alternative to oxygen. They leave the free nitrogen gas as a byproduct, and they close the nitrogen cycle. So what actually happens is, the wet soils or soils rich in organic matter, swampy soils, they release nitrogen in the form of gas. Nitrogen in the atmosphere, this closes the nitrogen cycle by the denitrifying bacteria. Basically, this is one perspective. Denitrifying bacteria which live deep in the soil, they use nitrates as alternative to oxygen, making a byproduct of nitrogen gas, and they release the nitrogen gas into the atmosphere or the environment, and here one chapter of the cycle gets closed. Okay, so now this can be divided into 5 stages. Here you have seen that I have discussed 4. One very important thing to understand is, which is the perspective of assimilations. We are the assimilators. So let us just see the microbiology and enzymology of nitrogen cycle of whatever we have seen right now. So the first step, I will be discussing whatever we have seen in 5 steps. The first step is nitrogen fixation, and I will be telling you the microbiology, biochemistry, and the enzymology behind that. So I would like to have your ears. Please pay full attention, and this is the interesting part of nitrogen cycle. Nitrogen, when it is fixed, it combines with hydrogen and with some electrons to give rise to ammonia. This is the reaction for nitrogen fixation. A greater amount of biologically available nitrogen is naturally generated via biological conversion of nitrogen to ammonia. Remember that thing? A huge amount of nitrogen which is available to the biological system, means for the biomass, it is brought about by this step, this reaction. Now this is carried out by a very specific small group of bacteria. They are very specific, and also cyanobacteria in the lakes, which are on the soil. There are azotobacter which are free living in the lakes. There are cyanobacteria which are capable of using the enzyme nitrogenase to break the bonds among the molecular nitrogen and combine it with hydrogen, as we have seen. So what is the enzyme which is responsible for breaking and combining the nitrogen? It is the enzyme nitrogenase. Is nitrogenase one enzyme? No, it is a cluster of enzymes. It is a multi-enzyme system. In the final lecture, I will be explaining about this nitrogenase and its molecular biology. So right now, just understand, all the microorganisms, they have an enzyme nitrogenase system. Nitrogenase is not one enzyme, it is an enzyme system which is capable of converting the atmospheric nitrogen to ammonia. Nitrogenase functions only in the absence of oxygen. This is very important to understand. The nitrogenase enzyme system, which is a product of NIF genes, NIF, nitrogen fixation genes. It is very sensitive to oxygen. It functions only in the absence of oxygen, and this absence of oxygen environment is maintained by certain very interesting ways, which you have to understand here. The very first step, nitrogen fixation, that is nitrogen combining with hydrogen and electrons to form ammonia, requires, this is fixation, requires NIF enzyme system, nitrogenase enzyme system. And you see the organisms, they are aerobic, they are highly aerobic, but the enzyme system is very sensitive to oxygen. So this exclusion of oxygen, or the exclusion of oxygen, or the, I am not saying anaerobic environment, try to understand. The organisms, they don't need an anaerobic environment except for denitrification. Here all is going out with respect, means if they have to combine oxygen molecule, how can it be in an anaerobic system? No, it is highly aerobic system, but the irony is that, or the dilemma is that, the enzyme nitrogenase is sensitive to oxygen. So this is carried out by the niche or the habitat of the bacteria at the first level. They live beneath the layers of oxygen, excluding slime on the roots of certain parts. There are, if you see Azotobacter, it is slimy, it has a slimy growth. Why that much? It secretes polysaccharide, and that polysaccharides nowadays are being used to produce bioplastics. Azotobacter, which is a free living soil bacteria, nitrogen fixing bacteria, capable of synthesizing nitrogenase, it has to protect its enzyme system for oxygen. How does it do? It lives beneath on the root, it secretes a certain slime like layer, and oxygen is excluded from entering the slime. That is one mechanism. That second mechanism is forming a heterocyst around its cell. It forms a heterocyst where the oxygen is excluded, means there is a intracellular diffusion of the nitrites and nitrates. It takes place, the nitrogen gas, this is diffusion takes place, active diffusion, passive diffusion takes place, but oxygen is excluded by the heterocyst structures. These are cell wall like structures. Okay, there are certain symbiotic bacteria like rhizobium. These rhizobium, they live in root nodules, and these root nodules are oxygen free zones. Means once they are present inside the root nodule, though they are aerobic, they have a oxygen free zone by which their fixation process is not affected by nitrogen. And on the roots of legumes and some other woody plants, the plant material itself supports the slime in Azotobacter or the non-diffusion of oxygen in that area. So by this way, by one way or the other, these organisms, they protect themselves from the atmospheric nitrogen. Doesn't mean they are anaerobic, they are just trying to protect their, they have evolved certain mechanisms like that, by which the oxygen doesn't really affect the nitrogenase enzyme. The second step is nitrification, the biological conversion of nitrogen in the soil from a reduced or a more oxidized state, and this is called as nitrification. What is nitrification? Nitrification is a stage where the biological, or it can be defined as a process where the biological conversion of nitrogen in the soil from a reduced state to a more oxidized state. Means in the reduced state it was combining with hydrogen, in a more oxidized state it will be combining with oxygen. And nitrification occurs in two steps. First, ammonia is oxidized to nitrite. This step is also called as nitrosification. Ammonia is oxidized to nitrite. Now this reaction, one, it converts ammonia to an intermediate. There is an intermediate formation called as hydroxyl amine, the hydroxyl amine, and it is catalyzed by the enzyme ammonia monooxygenase. I would like to hear you very carefully. This first reaction, it is catalyzed by the enzyme ammonia monooxygenase. Now this change is brought about by chemoautotrophic bacteria, which utilize chemicals as their energy source, and the prime names of the bacteria which can be taken here, which carry out this reaction nitrosification are Nitrosomonas, Nitrosolobus, Nitrosococcus, Nitrosospira. You can see their name predominantly starts with Nitroso, Nitrosomonas, Nitrosolobus, Nitrosococcus, Nitrosospira, Nitrosification or Nitrosification. Okay, so this change, that is the ammonia, which is getting converted into hydroxyl amine by the enzyme ammonia monooxygenase, is first part of nitrification. These bacteria, they obtain their energy by oxidation of ammonia, therefore they are chemoautotropes. Okay, chemoautotrophic bacteria, oxidation of ammonia to nitrite, NO2. Among the nitrifiers, Nitrosomonas are these all group of bacteria. Collectively they are called as Nitrosomonas, Nitrosobacter. Okay, so like that. These there are certain heterotrophs also. These reactions, that is ammonia to hydroxylamine, this reaction is also carried out by some heterotrophs like Streptomyces and Nocardia. What happens with this hydroxylamine? In the second stage, reaction 2, it converts this hydroxylamine to nitrite by the enzyme. I want your ears, all ears, listen carefully. Hydroxylamine oxidoreductase. Hydroxylamine oxidoreductase. This is the enzyme which is present in the second reaction by the same organisms. These are the nitrifying bacteria. Hydroxylamine is converted into nitrite. Now in the next step, nitrite is further oxidized to nitrate. The same group of bacteria, organisms, Nitrobacter, Aspergillus, Penicillium, Cephalosporium. This is the next step, that is the most important step, where ammonia is getting converted into a more utilizable form. So NO2, in the presence of oxygen, it gets converted to nitrate, NO3. So these two steps, both the steps, they can be classified as nitrification reaction. Nitrification of ammonia occurs in two steps, ammonia to nitrite. Now you see, when this is happening, oxidation states of the nitrogen are changing. Each compound is having a different oxidation state. What are those? We will see in lecture number 3. So two steps, ammonia to nitrite, nitrite to nitrate. I would like to just mention here, this is -3 oxidation state, -3 to +3, +3 to +5. Ammonia -3, nitrite +3. Nitrite +3 to nitrate +5. So -3 to +5 is the oxidation state. We will see that in detail. So this is the first stage, nitrification. The third step is assimilation. First was nitrification, second nitrogen fixation, second was nitrification, third is assimilation. Now what is assimilation? What we do? We eat the plant, meat, milk, egg, everything, all the stuff which is shown in this picture. Assimilation, we eat that stuff. Plants, they accumulate it, animals, they accumulate it. Assimilation is the process in which incorporation of the nitrites into biological tissues takes place. So in the form of nitrite, nitrate, ammonia, in whatever form it may be, it gets incorporated into our body. And plants, animals, we humans, we incorporate nitrate and ammonia formed through the nitrogen fixation into our body. It becomes a part of our biomass. The plants, they uptake it from the roots and incorporate into plant proteins and nucleic acids. We consume it and it becomes a part of our biomass in the form of proteins, amino acids, nucleic acids, etc. We then utilize the nitrogen through metabolism, and there are certain reactions again in our body where we secrete it as urea, urine, and again it forms a part of the nitrogen cycle. The fourth stage is ammonification. You can keep it in the second stage also, ammonification. It doesn't have a particular, as it is a cycle, it doesn't have a particular hierarchy. Ammonification is a process of hydrolysis of the proteins and other nucleic acids and other organic nitrogenous compounds into amino acids, that is proteolysis. Where does this take place? Now when it is assimilated, it is not a question whether it is getting ammonified in our body. Yes, it does, but it is not given back to the environment. As long as we are proteolysis is taking place inside our body, we will be excreting urea. Proteolysis or hydrolysis of proteins and nucleic acids and other organic nitrogenous compounds inside the plant or the animal or the cell takes place, it will be excreted as urea. That is one perspective. Second perspective is when the dead and decaying matter is added in the soil. There also ammonification takes place. Ammonification, recall, I have given a main perspective of ammonification of decomposition in the soil. So that is also ammonification. And daily all the living systems like animals, plants, insects, they are live, but they are excreting urea, or the defecation. Through defecation they are excreting nitrogenous compounds which is getting added in soil. That also is ammonification. So what is ammonification? In this, ammonia is formed. Okay, the first step in ammonification is hydrolysis of proteins, nucleic acids, etc. Amino compounds are then deaminated to yield ammonia. Then here you can see these amino compounds, they are deaminated to form ammonia. Ammonification usually occurs under aerobic conditions, and when under anaerobic condition, protein decomposition, it leads to conversion of ammonia to amines and its related compound also. So there is an aerobic one, there is an anaerobic one. Anaerobic system is called as anaerobic ammonia oxidation, or NMOX. That is one process which is utilized in the sludge sewage treatment, and it has found great significance in the treatment of sewage. But global perspective, the natural biogeochemical cycle, ammonification usually occurs in aerobic conditions and anaerobic conditions. The step is where ammonia is formed. Okay, ammonification, when it is anaerobic with respect to protein, as we see in sludge, when we see in decomposition, or when we see the compost, formation of compost, that is called as putrefaction. Degradation of proteins under anaerobic conditions to give rise to ammonia is called as putrefaction. This is also one important reaction in the nitrogen cycle. These amines are subsequently oxidized in the presence of oxygen to release ammonia. Almost all bacteria, actinomycetes and fungi, they can bring about proteolysis, and the amino acids produced are utilized for the growth of these organisms. All the bacteria, actinomycetes and fungi, they bring about proteolysis. Examples of ammonifying bacteria are Bacillus, Proteus, Clostridium, Pseudomonas and Streptomyces. So this is ammonification, where the amines are subsequently oxidized in the presence of O2 to form ammonia. There are various organisms responsible for this. The next and the last step is denitrification. So what is denitrification? This forms the final step of nitrogen cycle, and certain bacteria, they are capable of using nitrates as a terminal electron acceptor under anaerobic conditions. Now these are the redox reactions, and during this redox reaction, that is oxidation and reduction reaction, bacteria, they have a tendency to utilize the electron at the terminal stage, and on the basis of which, if it is oxygen, they are aerobic organisms, and those which have a capacity of some organic or inorganic compound apart from oxygen to accept the terminal electron, they are anaerobic bacteria. There are certain bacteria which are capable of using nitrates as a terminal electron acceptor under anaerobic conditions, and this is called as nitrate respiration. As a consequence of nitrate respiration, NO3 is reduced to N2 gas or nitrous oxide. Now this takes place in swampy conditions, as I said, beneath the ground, or wetland conditions, waterlogged soils, paddy fields. If you see the rice farms which need waterlogged conditions, it is completely waterlogged. So azolla, that is azotobacter and rhizobium, azorhizobium, these fertilizers, biofertilizers, they are added in waterlogged conditions because they carry out this nitrate respiration. So as a consequence of nitrate respiration, NO3 is reduced to N2 gas or nitrous oxide, and here it is then released back to the environment. This is called as denitrification. Denitrification leads to the loss of nitrogen from the soil. It is given back to the environment. It depletes nitrogen, and therefore it is not a desirable reaction from the fertility of the soil point of view. Fertility of the soil in terms of farming, I am saying naturally it may be okay, The escape of this molecular nitrogen into the atmosphere is also known as volatilization. Volatilization, that is volatility, due to its volatile nature, it is given back to the environment. Denitrification of the bound nitrogen to gaseous nitrogen is also carried out by many bacteria. There are certain denitrifiers, group of bacteria, which normally utilize oxygen as hydrogen acceptor in aerobic conditions, whereas nitrites and nitrates in the anaerobic conditions. Anaerobic conversion of nitrate into molecular nitrogen, which is called as nitrogen respiration and is a part of denitrification, is carried out by organisms like Pseudomonas. Then it is also carried out by Achromobacter, Bacillus, Micrococcus, etc. So you can see nitrate to nitrite to combining with molecular oxygen to give. So this is a reverse process, a complete reverse process, where nitrate to nitrite, nitrite to molecular nitrogen, and then atmospheric nitrogen. Okay, so there is a acceptance of electrons, protons, and the nitrogen gas along with water molecule. This water molecule, this is the one in waterlogged conditions, in swampy conditions, they prevail throughout the reaction, and this is the step of denitrification. As nitrates are used as source of electron acceptor, there is a net loss of nitrogen from the soil. This process is also called as dissimilatory nitrogen reduction, that is the loss of nitrogen from the soil back into the atmosphere. So if you see, as much as amount of nitrogen is fixed by the bacteria, that much amount of nitrogen is released back into the environment. So this process is called as dissimilatory nitrate reduction. Many soil bacteria like Thiobacillus denitrificans, they oxidize nitrogen, they also oxidize sulfur, these chemotropically, and they reduce the nitrogen to, that is nitrate to nitrogen. They are also sulfur reducers. That is one additional characteristic of this Thiobacillus species. So you can see this reaction, and ultimately nitrogen is given back to the atmosphere. So denitrification, if you see how many ways are there, that is N2O emission back to the troposphere, contribution, we saw how much is converted into the soil. Now this is a pie chart which is telling you how much nitrogen is given back to the atmosphere by which processes. Soils, they give back 6.6%, rivers 1.7% by the process of denitrification, oceans 3.8%, fossil fuels 0.7%, biomass burning 0.7%, and agriculture 2.8%. This is the general way by which nitrogen is given back or lost into the environment. The general pathway of denitrification, if you see, nitrate is first reduced to nitrite, which is then transformed to nitrous oxide. All these reactions involve microorganisms and their enzymes. The nitrous oxide is converted to nitrogen with NO2 as an intermediate. So if you see HNO3 to 2HNO2 to 2NO to NO2 to N2, this is in 4-step reaction. The enzymes involved in conversion of HNO3 to HNO2 is nitrate reductase. Nitrate reductase. HNO2 to NO, second enzyme, that is nitrate reductase. Nitrite reductase, nitrate reductase. To NO to N2O is nitric oxide reductase. All these are reductase group of enzymes carrying out the reduction reactions, and the third N2O to N2 is given by nitrous oxide reductase. These are the 4 enzymes which are capable of converting nitric acid to nitrogen gas, releasing it back to the atmosphere. So this is a reverse of nitrification process, that is reduction of nitrate to nitrite and then to ammonia. As the organisms, they are able to obtain the cellular nitrogen through ammonia assimilation, the process is also called as assimilatory nitrate reduction. So this is the last part of the nitrogen cycle, assimilatory nitrate reduction. So are there other ways by which nitrogen can be returned to the atmosphere? This should be the final question. We have seen the bacteriological perspective, how the microorganisms, they are involved in the release of the nitrogen to the atmosphere. There are other ways by which nitrogen can be returned to the atmosphere. Emission from industrial combustion and gasoline engines, they create nitrous oxide, that is the automobile pollution, a sort of emission from industries, combustion reaction which is there, gasoline engines which create nitrous oxide gas N2O, it is released in the environment. Naturally, when there is volcano eruption, when there is a volcanic eruption, it emits huge amounts of nitrous oxide gas. But these are very rare phenomena, not continuously going on. But when it occurs, there are some volcanoes which are continuously going out in the nature. There are live volcanoes which have been live for years together, so they are continuously contributing N2O to the atmosphere. From the bottom of the earth, where there are sediments of nitrogen, these are at high temperatures converted, the bonds are broken and nitrogen gas is released into the atmosphere in the volcanic reactions. So in general, this is all an overview of the nitrogen cycle. As you can see, the first stage here is nitrogen fixation. Nitrogen in the atmosphere is converted to nitrites or ammonia. The second stage, you can see, this is, as I said, a perspective, second, fourth, wherever you can keep it, it is a part of the cycle. Ammonification, where the legumes and the soil bacteria, they fix it. Decomposition, that is ammonification, decomposition, it converts the dead material into ammonia. The nitrogen fixation by the leguminous plants, by symbiotic and soil bacteria, free living by non-symbiotic. Then ammonification, decomposition into ammonia. Then nitrates are added into soil, ammonia is converted into nitrites and nitrates, a stage called as nitrification. Then the nitrates are converted into denitrification back into atmospheric nitrogen. So this is the stage of denitrification. Overall, this is nitrogen cycle. Now one very interesting part of nitrogen cycle is the genetic system involved in this. These are called as the NIF genes. All these organisms which you saw the microbiology, the biochemistry and the enzymology of nitrogen cycle. Now there is one small perspective which is called as the genetics of nitrogen cycle, or the molecular biological perspective of the nitrogen cycle. In my next lecture, the final part of this nitrogen cycle, we will see the molecular biology of nitrogen cycle. I hope you are understanding this very interesting and beautiful natural cycle in the nature, which is carried out exclusively by microorganisms. Remember, if microorganisms were not there, we would have not got nitrogen. If you have not got nitrogen, you would not be able to synthesize, forget proteins or forget anything. You wouldn't have a DNA, and if you didn't have a DNA, you wouldn't have survived. Not a single biomass species on the earth would have existed. So these are the key players in the biogeochemical cycle. All biogeochemical cycles, when we are studying nitrogen cycle, I am emphasizing on nitrogen cycle. Similarly, when we will be seeing phosphorus, sulfur, etc., these microorganisms, they will be key players without which the transformations do not take place. The recirculation of these elements, taking from the environment, giving back to the environment, in the middle converting it into various forms which we need for life, living on the earth for some duration of time, dying off, getting converted into another forms, then again forming some other biomass. This is a cycle which is going on for years together, millions of years together, and nitrogen is one very important link in the chain. Okay, so with that I end my lecture here. In my next lecture, we will see the molecular biological perspective of nitrogen cycle, special reference to the NIF genes, the genetic systems and the redox oxidation states of nitrogen. Till that time, goodbye, happy studying, get more knowledge, become more informative, become wise and smart. Goodbye.